Hello everyone, my name is Visalhounuo. In this video, I'll be explaining on acid-fast staining. It is a method to stain acid-fast bacteria like Mycobacterium slender rod bacilli. They do not stain readily, but once stained, resist decolorization due to the presence of lipid-rich, waxy materials, that is, mycolic acid on the cell wall. So they are called acid-fast bacilli. There are two methods of staining acid-fast bacteria. One, hot staining method, example, ZIELH-NEELSEN staining. Second, cold staining method, example, Kenyon staining. The principle of ZIELH-NEELSEN stain is, when the smear is stained with carbylfuchsin, it solubilizes the lipoidal metal present in the mycobacterial cell wall, but by the application of heat, carbylfuchsin further penetrates through the lipoidal wall and enters into cytoplasm. Then after all cell appears red. Then the smear is decolorized with decolorizing agent, but the acid-fast cells are resistant due to the presence of large amount of lipoidal material in the cell wall, which prevents the penetration of decolorizing solution. The non-acid-fast organism lacks the lipoidal material in the cell wall, due to which they are easily decolorized, leaving the cells colorless. Then the smear is stained with counterstain methylene blue. Only decolorized cells absorb the counterstain and take its color and appear blue, while acid-fast cells retain the red color. The requirements for the test are glass slide, spirit lamp, staining rag, Pasteur pipette, and microscope. The reagents required are: primary stain is strong carbylfuchsin, decolorizer is 20% sulfuric acid, counterstain is methylene blue. Coming to the procedure, place the heat-fixed smear onto the staining rag and using a Pasteur pipette, flood the smear with carbylfuchsin. Then heat the slide by passing the flame of the spirit lamp below the slide till fumes arises and keep it for 5 to 8 minutes. After which, gently wash under running tap water. Then decolorize with 20% sulfuric acid for 1 to 2 minutes until the smear is sufficiently decolorized. Then place the slide back to the staining rag and flood the smear with methylene blue and keep it for 2 minutes. Then gently wash under running tap water and air dry the slide at room temperature and observe it under the microscope at 100x oil immersion. Under the microscope, the acid-fast bacilli get stained by the strong carbylfuchsin stain and appear red in color, while the past cells and inflammatory cells get decolorized and retains the counterstain methylene blue, so appears blue in color. Examples of acid-fast bacilli are Mycobacterium tuberculosis and Mycobacterium leprae. And this is the grading of sputum smear or AFB grading. The number of AFB per 100 fields, if it is 0, then the result is negative. 1 to 9 AFB per 100 field, this grading is scanty and resulted positive. 10 to 99 AFB per 100 field, grading is 1 plus, result is positive. 1 to 10 AFB per field, observed in 50 field, grading is 2 plus and result is positive. More than 10 AFB per field, observed in 20 field, grading is 3 plus and result is positive.